Hello, everyone. Welcome to BioNow, the weekly webinar series where we discuss health, wellness, and longevity. I'm your host, Simran. In this week's webinar, we will explore how technology, when integrated with molecular science, can do wonders for all. Our speaker for this webinar, Dr. Preethi Balasundaram, is a scientist specialized in bioinformatics and has an extensive background in research. She pursued her Ph.D. in computational biology and bioinformatics with expertise in drug resistance analysis, molecular simulations, and drug repurposing methods. From working as a postdoctoral fellow at University of Lethbridge to being a research fellow for a lung cancer project, her journey has just been commendable. And today, she will help us in exploring how artificial intelligence, apart from helping you find your pet's name, can actually help to do innovations in science. But before we proceed, I would request all the participants to adhere to the following rules. First, please keep your microphone muted at all times, as background noise can be disruptive. Second, use the chat box if you have any concerns or questions or face any technical issues. We also want to inform that this webinar will be recorded and shared on various social media platforms for learning purposes. Also, with the aim to continuously improve, we would really appreciate your feedback, so please feel free to share it with us at the end of the session. Without further ado, please join me in welcoming Dr. Preethi Balasundaram to learn something exciting today. Over to you, Dr. Preethi.
Thank you, Simran. Uh, Good evening, everyone. Everyone, I shall be talking about integrating omics to AI. I just want to thank you, everyone, for attending. We are seeing tremendous improvement in the technology and also in the AI. Of these upcoming term of artificial intelligence is a buzzword for last few years and this fast-paced world. Before we understand what is integrating omics to AI, let's understand what is AI. So now what is mean by artificial intelligence? Artificial intelligence is a computing concept that helps a machine think and solve complex problems as we human do in our intelligence. In simple words, it is a training machine to intimate or reproduce human task. The term robot was first coined in the year 1923. Then in 1946, foundation of neural network was established. John was the first person to coin the term artificial intelligence and he ran the program using that. And to solve the problems, AI can utilize different methods such as machine learning, deep learning, artificial neural networks, and so on. Machine learning is a data parsing technique to study the data and apply that information to deliver decision. For example, YouTube suggestions and Amazon product suggestions are based on machine learning approach. Let us look at the deep learning method. Well, deep learning is the subfield of machine learning technique. It structures the algorithm in layers to create artificial neural network that can autonomously study the data and make intelligent decisions. The structure and functions used in it are trained like human brain. For example, autonomous vehicles or virtual assistants are the examples of deep learning. Presently, AI is implemented in all the fields. I would like to discuss about drug discovery process as an example. Before jumping on to the AI in drug discovery, we need to understand about the stages of drug discovery process. Drug discovery can be divided into 4 phases: discovery and development, preclinical studies, clinical studies, and market approval. In the discovery and development, we are focusing on different sections. In that, target identification and validation is the first step. Target is a cellular or molecular structure which is responsible for a disease. Target can be enzyme, receptor, hormone, nucleic acid, or ion channel. We can use different omics data to identify the target for identification. such as genomics, proteomics, transcriptomics, metabolomics, and so on. Just quick overview of what is omics. Genomics provides an overview of complete set of genetic information provided by DNA, while transcriptomics looks into the gene expression patterns. Proteomics studies about dynamic protein products and their interaction, while metabolomics is also an intermediate step in understanding organisms' entire metabolism. And then what is epigenomics? Epigenomics explores heritable, reversible modification of DNA and chromatin that do not change primary nucleotide sequences. How we can integrate AI? There are different AI-based workflows exist to integrate different omics data for target identification. Drug targets must be identified and positioned correctly to manufacture a new drug. The prediction of protein target can be achieved by using different AI techniques: network based, genetic algorithm based, and neural network based, decision tree, and so on. And a few companies are working on different AI techniques. For example, PandaOmics is a tool was designed by in silico medicine company to integrate omics data for drug discovery. Afterwards, once the target is identified from the omics, what we should do? Once the target is identified, we will search for the three-dimensional structure of the target molecule from the publicly available database, such as protein data bank can be used for the protein data 3D structure of the protein. If structure is not available in the database, it can be generated with the different tools such as Modeler, Swiss Model, iTaser, and so on. Modeler and Swiss Model, which uses comparative structure modeling technique, that is, which predicts the 3D structure by aligning the target protein to one or more known protein structures, whereas iTaser uses protein hierarchy to generate the structure. And SciSpread and AlphaFold 2 are the recent examples of AI-based tool which utilize the machine learning and neural network algorithm to predict the protein structure, 3D structure of the proteins. Now, coming to lead identification. New chemical compound can be identified by different ways. Ritual screening or random screening of compounds with a similar structure or properties against the chemical databases. The figure displayed here, the R represents the drugs which are similar in gene expression from one family of drugs. Blue colored drugs are belong to the one family of drug which is screened for similar gene expression profile against databases, and the orange colored drugs has the similar gene expression with the family of drugs which has the connection with one or more drugs in the same family. Then we can also target the drug or identify the drug using drug repurposing and drug repositioning strategies. Let us talk about what is drug repurposing and repositioning strategies. Recently, drug repurposing and repositioning strategies were used to identify new therapeutic indication of old drugs or withdrawn drugs in order to reduce the processing time of the drugs. Repurposing an active drug that is already on the market for new disease by targeting the new targets. Using withdrawn drugs can be used for new disease for its new indication. For example, thalidomide. Thalidomide is a well-known example for the repurposed drug. It is withdrawn for its adverse activity which affected thousands of victims worldwide in the 1980s. This drug is repurposed for its first-line treatment for melanoma patients. Here are the few publicly available database in that we can identify new leads and then we can process further. Database such as PubChem, ChemBL, and Drug Bank can be used for lead identification. Different tools such as DeepChem, Deep Neural Network, QSAR or Deep Neural Net QSAR are Python-based AI tools to determine the molecular activity of the drug molecule or the lead molecule. For a drug to be in the market, it should come across all the ADME properties. So deep tox can be used as a toxicity predictor, which predicts the ADME properties of that particular molecule. Organic tool can design and generate the molecule with the desired properties, whatever we want as a drug. And neural graphic fingerprint can predict the properties of the chemical molecule using neural network algorithm. Then, once the target is identified, we should go forward with molecular docking to dock the protein and the ligand molecules for its activity. It's a key tool. Molecular docking it's a key tool in the structural molecular biology and computer-aided drug design. It is used for the virtual screening, binding affinity, and binding free energy calculations, and also for visualizing various types of interactions between the molecules. This figure represents the interaction between protein and the drug molecule, and the dotted lines represents the hydrogen bond interaction between them. From this figure, how the drug is binding to that particular protein, and their hydrogen bond represents how well it is binding to that particular protein. Then, here are the few examples of molecular docking tools. Schrödinger Glide can predict the protein ligand binding score and rank accordingly. It can process about thousands and thousands of lead molecule at a time. Autodoc and Autodoc Vena are based on Python-based to predict the protein ligand binding and affinity. Potential net can estimate protein binding and ligand affinity using neural network algorithm. Then once the protein and ligand is bind together, then we should check the stability of that protein and the ligand. And this protein and ligand stability can be detected by molecular dynamics simulation. The stability of the protein and the drug can be analyzed using molecular dynamics simulation studies. It has become interestingly useful in modern drug development process. It predicts the structural, dynamical, and thermodynamical properties of the molecular system at the atomic level. This RMSD figure, first figure, represents the RMSD, which gives you information about the protein drug stability over the time period. Whereas second figure, RMSF, which provides the information about the atom stability throughout the time period. And there are few examples of molecular dynamics tools, that is CHARM, AMBER, GROMACS, NAMD, LAMPS are used as a molecular dynamics tools to predict the thermodynamical properties and structural and dynamical variations in the protein drug or either protein compound for their stability. Few AI-based features are incorporated in the mind-sponge simulations. Once the molecular dynamics is done, then what we should do? So once the properties computational work is done, the researchers will identify a promising compound for the development, and they will conduct experiment to gather the information on how the drug is absorbed, distributed, and metabolized and excreted in the body, and its potential benefits and mechanism it's illustrated. The best dosage to be administered, the best way to give the drug either by mouth or injection. What are all the side effects or adverse effects that can often be referred to as toxicity, which may affect the human. How it is affecting different groups of people, such as gender, race, and ethnicity. How it can interact with the other drugs and their treatments. Its effectiveness as compared with similar kind of drugs, that is same family of drugs. This can be achieved by preclinical studies. Before testing the drug in people, researchers must find out whether it is potential to cause serious harm, and it can be analyzed using toxicity analysis. First, we will for preclinical studies, the researchers will go for in vitro studies. In vitro studies are exposing the target cells to the novel drug and monitor its side effects. This helpful for identifying whether or not novel drug has toxicity or carcinogenic effects on the cells. Then they will go for in vivo testing. In vivo testing is testing the novel drug molecule on the living organism or the whole cell to evaluate or more accurate effects on drug molecule in the complex model. For example, you can test the drug molecule on rabbits or mouse models, that is called in vivo testing. Once the preclinical studies is done, we should go for clinical studies. Preclinical studies are not very large, however, these studies will provide detailed information about dosing and toxicity levels of the drug. Then followed by the clinical studies. This clinical studies has 4 phases. Each of the phase is crucial and leads to the next. Phase 1, we will test the drug with 20 to 100 healthy volunteers and see the effect of the drug in the volunteers. If it has an adverse effect in healthy volunteers, they will stop the studies and they will move forward. Phase 2 will confirm with the several hundreds of volunteers with the disease condition. Phase through once the drug is passes through the disease condition, they will go forward with 300 to 3,000 volunteers with the disease condition. Phase 4 will be the several thousands of volunteers with the different ethnic conditions, and male female gender variations will be there. With that, they will confirm with the drug effectiveness, either it has any adverse effect is there, they will stop taking the drug, moving forward to the FDA review. Once the drug passes through all these phases, it will comes to the FDA review. What is FDA review? Like new drug application, like once the FDA is approving a drug, for that they need all the information about the all the clinical trials, preclinical trials, what are all the data they are having. They will submit to the FDA, and we need approval for the drug to be marketed. A new drug application will tells you the story about the drug. It is a purpose to demonstrate the drug is safe and effective for intended use in the population studies. Once the basic question of drug safety is answered, FDA inspectors will review the reports on all the studies and data analysis. The FDA inspectors might be medical officers and statisticians will review the clinical data, while the pharmacologists review the data from the animal studies. If the drug is proved to be safe and effective for its intended use, then it will be processed for labeling and marketing. Here are leading pharmaca companies and partnered AI organizations for the drug development. Roche has partnered with Recursion Pharmaceuticals and Okin for the new drug development. And few other companies, other than listed companies, few other companies like Google, DeepMind, In Silico Medicine, Deep Genomics, Helix, etc., are also making huge investments on AI-based drug discovery. And there are few drugs which is under AI-based drugs are under clinical trials. We can expect them in the market very soon. Artificial intelligence is not a suitable it's not a substitute for human intelligence, but it is a tool to amplify the human creativity. And thank you everyone for joining.
Thank you so much, Dr. Preeti. We're grateful for you sharing your wealth of knowledge with all of us. It was such an informative session, and I'm certainly more informed and aware about the role of AI integration into the modern development and drug discovery. So, one thing that we will do right now is Dr. Preeti will now take questions from the audience on the discussion that we had today. Please use the chat box for asking the questions or request to unmute if you would like to use your microphone. So we have a first question. So, one, how genomics can help with clinical trials?
Genomics will help. Yeah, just to answer the first question, genomics might help in target identification of that particular um, target identification for the particular protein, so that genomics will be helpful for getting the target needs to be identified for that particular drug. So in this way, genomics will help in the clinical trials and those things for the drug discovery.
So we have another question. I assume that all the AI tools presented focus on the human proteins. Are there AI tools available for the microbial proteins or microbiome therapies?
Yeah, there is a lot of AI tools. I just described few of them, and there are more microbial proteins based AI tools are there for different sections. So it can uses machine learning, deep learning, and other neural network based tools are available for microbial proteins.
Perfect. There are so many AIs. Actually, I have a question for myself, right? So, given that we touched bases about genomics and how it helps with the clinical trials, what about AI for our genes, right? We have so many of them and they're so diverse. So it's very hard to create a profile manually. What do you think a future association AI with genetics and how it will grow?
Yes,
I mean, just a
actually,
speculation.
yeah, like actually, most of the companies are working on reducing the just analyzing our genetic panels to based on AI, how AI can read the genes. Most of the lots of AI based companies are working on it. Actually, there are a few other tools and algorithms are already developed.
Wow. Well, that's very insightful. And we have one more question. Um, can you provide any examples of the
Actually,
invention?
we don't like currently they are in clinical trial. As I mentioned it at the last, mostly everything are in clinical trials. We don't know the exact name of it. Few drugs are in the phase 4 trials. We can expect them very soon in the market.
That's incredible. Just thinking about how something that you can create can help you in the future with simplifying a lot of tasks, such
Yeah,
as suggesting.
it's actually yeah.
We have a question. AI will replace humans in the future?
Might be. Because
My,
AI can't replace human. It is trained to do job or think like human. AI can't replace it.
yeah, that's something that I agree
Which
also.
one? Yeah, because humans are training machines to do that. So
Exactly. I
yeah,
believe that humans have a creativity
it's
and then
creativity. Yeah,
AIs are just helping with the certain
yeah.
tasks.
Whatever we are designing as a programmer, or how we train the machine to analyze the data, that is what we actually we are creators.
Yeah,
Yeah,
can't agree more. We have one more question. How
yeah,
does combination of AI and genetic data improve personalized health recommendations?
yeah. Because AI is combined with the genomics so that it can now sequencing has become common in every area. So we can have the genomic data then and then, and it can predict their personalized genome, and then we can train AI algorithm to have a personalized medicine based on like their ARG, like antimicrobial resistant profiles, how they are resistant to some other drugs or so. We can train them, and then we can use it for the personalized healthcare or personalized medicine.
Wow, a lot of AI discussion, especially probably after the ChatGPT release.
Yes.
People tend to integrate it more and more into everyday life.
Yeah.
So pretty hot
Nowadays,
topic, amazing talk actually, an amazing topic, Dr. Preethi. I'll give people some more time just to maybe finish typing or thinking about their question. We have a lot of appreciations for the awesome presentation. So I want to share all those appreciations with you, Dr. Preeti. The talk was actually amazing
thank you.
and we learned a lot today. Okay, that brings us to the end of this webinar. Thank you for your participation and all your questions. I would also like to take time out and thank to all of you, our virtual audience, who joined us today from within Canada and around the world. It's amazing to see how the diverse group of people come together to explore science. Before we officially wrap up, I would like to take a moment to express my gratitude to you, Dr. Preeti, for taking your time out and it's providing us with this informative session. We appreciate your generously sharing your expertise and knowledge in the integration of omics to AI and how to develop and discover drugs with the help of AI. I encourage you all stay curious, keep learning, and join us on the 20th of December for a podcast from our amazing dietitian, Dr. Raman Kapoor. Please remember the more we know, the better we are equipped to make informed decisions to improve our lives and the people around us.